Hello everyone, today we are going to discuss about photosynthetic unit and pigment system. So photosynthesis process in plants usually encompasses two processes. One is light reaction and second one is carbon fixation reaction or carbon assimilation reaction. So in these light reactions, the photosynthetic pigments absorbs light and transduce it into the chemical energy, that is ATP and NADPH. In this process, they also evolve some oxygen molecule. Okay. And in carbon dioxide fixation reaction, this ATP and NADPH is used to reduce carbon dioxide to starch or sucrose or glucose and some other byproducts. So these chlorophylls or photosynthetic pigment used in the photosynthesis process usually act as a unit to produce oxygen molecule or to do this photosynthesis process. In 1932, Robert Emerson and William Arnold found out that 2400 chlorophyll molecules acts as a unit to produce oxygen molecule. Okay, 2400 chlorophyll molecule acts as a unit to produce one oxygen molecule per 8 photons absorbed. And this 2400 chlorophyll molecule acts as a unit. That unit is known as photosynthetic unit. And this photosynthetic unit size varies from species to species, and the size varies from 300 to 5000 chlorophyll. The size varies from 300 to 5000 chlorophyll molecule per species to species. Varies in different organisms. Now, in some photosynthetic process, this oxygen molecule evolves or releases, but there is also some photosynthetic organism which does not release oxygen. So those photosynthetic organism which releases oxygen are known as oxygenic photosynthetic organism. And those photosynthetic organism who does not release oxygen, they are known as non-oxygenic photosynthetic organism. Okay. Now, till 1930, researchers used to consider this carbon dioxide. As we know the photosynthesis formula, carbon dioxide, water gives rise to, in light and chlorophyll, it produces form of starch, water and oxygen molecule. This is the basic photosynthesis process or formula or reaction we know. But what happens in non-oxygenic photosynthesis? This oxygen does not evolve. Till 1930, researchers used to consider this carbon dioxide as the source of this oxygen molecule. But in 1930, C.V. Van Til from Stanford University, he found out that there is a photosynthetic bacteria named as Chromatium vinosum, who does carbon dioxide assimilation, who assimilates carbon dioxide but without releasing this oxygen. So in 1930, C.V. Van Til found out that there is a bacteria, photosynthetic bacteria, Chromatium vinosum, which does not release oxygen, but it does assimilate the carbon dioxide. So here, this is the example of non-oxygenic photosynthetic organism. So in this non-oxygenic photosynthetic organism, these bacteria uses H2A, hydrogen sulfide, H2S, for the electron donor, as an electron donor, and it produces sulfur instead of oxygen. So here, when we look into the non-oxygenic photosynthesis process, it will be carbon dioxide with some hydrogen sulfide in presence of light and chlorophyll molecule, that is bacterial chlorophyll molecule, produces starch, water and sulfur instead of this oxygen molecule. Okay. Now we say that here we can understand this carbon dioxide is not the source of oxygen, because here is also carbon dioxide present, but there is no evolution of oxygen. Here we are getting sulfur. That means he said challenged this thought of the previous researchers who used to think that carbon dioxide is the source of oxygen. He challenged this concept and he found out that there is this oxygen cannot be produced from the carbon dioxide. Later on, in 1937, there is a scientist called Robert Hill who gives a theory that is known as Hill's reaction. In 1937, Robert Hill gives a theory or proposes a theory regarding this oxygen molecule evolution that is known as Hill's reaction. So he did an experiment with isolated chloroplast. He takes some isolated chloroplast and this chloroplast lack those necessary enzymes required for the carbon dioxide fixation. Okay, he took some isolated chloroplast and with some oxidants. Okay, oxidants mainly ferrocyanide, ferric oxide and DCPIP. So this DCPIP is also known as Hill's reagent. This is 2,6-dichlorophenols. So it's a non-physiological oxidant. So he took isolated chloroplast without the enzyme for carbon dioxide fixation with some oxidants and he got NADPH, hydrogen atom and oxygen molecule. Okay, he get that there is NADPH produced, one hydrogen atom is produced and oxygen is evolved. So now he stated that this oxygen is the production of photolysis of water, not from the carbon dioxide, because there is no enzyme to fix the carbon dioxide inside it. That's why he states that the carbon dioxide is not the source of oxygen in photosynthesis process, rather oxygen is evolved from the photolysis of water. So this is known as Hill's reaction. Okay. Then we'll discuss about pigment system. So now we'll discuss about pigment system. Pigment system is also known as photosystem, and these pigment systems are found in oxygenic photosynthetic organisms. So all oxygenic photosynthetic organisms possess pigment system. There's two pigment system, but this pigment system came into light because of two phenomena. So first we will discuss about that two phenomena. One is red drop phenomenon. Red drop phenomenon, and the second one is Emerson enhancement effect. Emerson enhancement effect. So these are the two phenomena which now we are going to discuss about. So first, this red drop phenomenon. In 1943, Robert Emerson and Carlton Lewis performed an experiment on chlorella, a green algae. So he performed that he tried to study the effect of visible light spectrum on the oxygen evolution of the photosynthesis process. So he studied the effect of visible light spectrum on the quantum yield of photosynthesis. So what is this quantum yield? Quantum yield is the number of, this quantum yield means the number of oxygen produced per photons absorbed. Okay. To understand it in a better, we will plot a graph, in which graph we will take x-axis for the wavelength of light, and for y-axis we will take quantum yield of photosynthesis. Okay. So as we know that visible light spectrum ranges from 380 nanometer to 750 nanometer. So we will take 400, 500, 600 and 700 nanometer wavelength of light. And for quantum yield we will take 0.05 and 0.1. Now we will see what they have observed from the absorption spectrum and quantum yield. So first they saw the absorption spectrum. So absorption spectrum is constant, at 500 nanometer it is slightly deep, and at 680 nanometer it falls off drastically. It immediately falls off. Okay, and this is the absorption spectrum. Then we look for the graph of quantum yield of photosynthesis. So yielding of photosynthesis or evolution of oxygen was somehow constant till 500 nanometer, then it's a slightly dip, then it again rises, and at 680 nanometer it falls off drastically. Okay, so this is the point of 680 nanometer. 680 nanometer is the wavelength of far red light. So now we can say that this far red light, this quantum wave, that means oxygen evolution is very less or it falls drastically. That means this far red light is not effective for the photosynthesis process. Now as it is dropping in the red part or the far red region, that's what is known as red drop phenomenon. Okay, and there is a smaller dip at 500 nanometer. So now the question is, why there is again dip at 500 nanometer? Because this shows the lower efficiency of photosynthesis due to the light that is absorbed by the accessory pigment carotenoid. So these light are absorbed by carotenoid, and this shows the lesser efficiency of photosynthesis, lesser efficiency of this light to do photosynthesis process. So this is red drop phenomenon. Now again, Emerson and his colleague again said another experiment where they applied two light beam. One is red beam of light, that is less than 680 nanometer. Another one is far red light, that is more than 680 nanometer. So first you applied single light for the photosynthesis process, and also to understand it better we will plot a graph here again. X axis stand for the time and y axis stand for rate of photosynthesis. So here, when first they applied the far red light. When they applied far red light, when the light is on, the rate of photosynthesis goes up. At some point it becomes constant. Then they switch off the light and it comes down. Okay. Again they applied red light. Here also the photosynthesis goes up. At some point it becomes constant. Then when the light is off, it becomes down. But at last, they used both the lights simultaneously. They applied both the lights simultaneously and saw that the rate of photosynthesis is 2 to 3 times greater than the sum of the rate of photosynthesis obtained separately. Okay, let's consider this is far red light, this is red light, and this is when the both far red and red light was applied. And here the rate of photosynthesis is 2 to 3 times greater than this, the sum of this. And this is known as Emerson enhancement effect. It shows the effect of both the light to enhance the rate of photosynthesis, and this process was carried out, or the experiment was carried out by Emerson. That's what is known as Emerson enhancement effect. So now we have discussed about the red dot phenomenon and the Emerson enhancement effect. From red dot phenomenon we understood that far red light is inefficient for photosynthesis process, and we studied the effect of visible light spectrum on quantum window photosynthesis. In Emerson enhancement effect, we understood that when red light and far red light, both beam of light was applied simultaneously, the rate of photosynthesis is 2 to 3 times greater. So these are the two phenomena. Now we will discuss about the pigment system. So as I said, pigment system is mostly seen in all the oxygenic photosynthetic organism. So in oxygenic photosynthetic organism, there are two types of pigment system. Okay. So in pigment system, what happens? In all photosynthetic organism, there is photosynthetic pigment. So these photosynthetic pigments bound with some protein and produces protein pigment complex, and these protein pigment complex are known as pigment system. So first we'll discuss about a basic structure of pigment system. This pigment system, the basic component of pigment system, there are two components of pigment system. Pigment system is also known as photosystem. So there are two components of pigment system. One is reaction center or photochemical reaction center. Another one is antenna molecule. Okay. One is reaction center, another one is antenna molecule. So this reaction center, what happens when the light energy is absorbed, then it is sent to reaction center. Reaction center contains a special pair of chlorophyll A which conserves the light energy and it transduced into the chemical energy. So that's why it is the photochemical reaction center. Here photochemical reaction center is of two types. Again, one is type 1, another is type 2. Type 1 is a APase type reaction center. Type 2 is a pheophytin quinone type of reaction center. Okay, pheophytin quinone type of reaction center. And here in antenna molecule, it is also known as light harvesting complex. As the name suggests, light harvesting complex means it harvests the light. It absorbs the light and then immediately efficiently sends it to the reaction center for further photochemical process in which this light energy transduced to chemical energy. So this light harvesting complex is of two types again. One is core antenna molecule, another one is peripheral antenna molecule. Core means outer antenna molecule, peripheral means core means inner antenna molecule, peripheral means outer antenna molecule. So in light harvesting complexes usually contains a 7 molecule of chlorophyll A, 5 molecule of chlorophyll B and 2 accessory pigments. Okay. In general, light harvest complex contains 7 molecule of chlorophyll A, 5 molecule of chlorophyll B and 2 accessory pigments. This number of light harvesting complex varies from different organism, that is from organism to organism or species to species, that is 200 to 300 pigments. Okay, and this light harvest complex are present in almost all photosynthetic organism. One is cyanobacteria, exception there is cyanobacter and red algae. They use this light harvesting complex, but not any of these light harvesting complex. They use phycobilin as their light harvesting complex. Okay, and there is another exemption. One is Helicobacter, who does not use any light harvesting complex. So Helicobacter does not use any light harvesting complexes. So this is the component of photosystem. So in this oxygenic photosynthetic organism, there are two types of photosystem or pigment system. They are known as pigment system 1 and pigment system 2. Okay, so first we will discuss about pigment system 1. It is, as we know, that it is a protein pigment complex, and pigment system 1 actually contains APase type of reaction center. Okay, its reaction center is APase type reaction center with light harvesting complex 1. Okay, it contains APase reaction center and light harvest complex 1. Along this, beside these two reaction center and antenna molecule, it also contains some proteins, chlorophyll A molecule, carotenoid and phytoquinone and some other factors. Okay, so all this reaction center contains a special pair of chlorophyll A which can transduce the light energy to chemical energy. And here the reaction center, the chlorophyll A can absorb the light energy of wavelength 700 nanometer. That's why it is known as P700. Okay. And this PS1 usually uses the light to transfer the electron from plastocyanine to ferroxide. Okay. Now we have seen the reaction center of PS1 is APase type with the light harvest complex 1. Then we have seen the other factors. It usually contains protein, chlorophyll A, carotenoid, phytoquinone and some other factors. Then the specialized chlorophyll can absorb 700 nanometer of wavelength of light. That's it is known as P700, and it is usually found in the on-stacked resin membrane of stroma and grana thylakoid of chloroplast. Okay, so it is found in the on-stacked membrane of stroma and grana thylakoid. So this is about the photosystem 1. Okay, now we will discuss about photosystem 2. As we know that all photosystem is a protein pigment complex. So both 1 and 2 are protein pigment complex. One reaction center is APase reaction center. So the type 2 reaction center is pheophytin and quinone type of reaction center. So it causes pheophytin and quinone type of reaction center with LHC2, light harvest complex 2. Along with this, it also contains two membrane protein. Okay, two membrane protein, that is D1 and D2. These proteins are almost like 39 kilodalton each. Okay, and also with it contains some other proteins like cytochrome B559, some chlorophyll A and other factors. Okay, Along with this reaction center and an antenna molecule, it contains some two membrane protein D1, D2 with some proteins like cytochrome B559 and chlorophyll A molecule and some other cofactors. The specialized chlorophyll A pair of the reaction center can absorb efficiently the light of wavelength 680 nanometer. That's why it is known as P680. Okay, it is known as P680. It is known as P700. It is found in the stacked region of granathylakoid. So PS2 is found in stacked region of granathylakoid. Now we have understood this two pigment system. We have understood the basic difference between two pigment systems. One is the reaction center. Pigment system 1 has the reaction center FES type. Pigment system 2 has the reaction center phyophytin type. Along with this, it contains proteins, chlorophyll A, carotenoid, phytoquinone and other factors. PS2 contains two membrane protein D1, D2. Then proteins like cytochrome B559 and chlorophyll A molecule. It can absorb 700 nanometer of wavelength of light, that's why known as P700. It can absorb 680 nanometer of light, that's why it is known as P680. It is found in the on-stacked region of stroma and granathylakoid, and PS2 is found in the stacked region of granathylakoid. Now the ratio of the number of PS1 to PS2 is PS1 photosystem 1 to photosystem 2, the number ratio is 1:1.5. 1:1.5. Okay, and this number can change, depending upon different light conditions. So in today's class we have discussed photosynthetic unit. Then we have discussed Hill's reaction, oxygenic and non-oxygenic photosynthetic process. Then the two phenomena, red dwarf phenomenon and Emerson enhanced effect. Then the components of photosystem or pigment system and two pigment systems, pigment system 1 and pigment system 2. So if you have any doubt regarding today's discussion, please comment down below. Thank you.